now have Sioux City, Iowa, a Mission 7 experiment. This is a flight experiment, grade 9.
Hello, I'm Rachel, this is Robert, and this is Tammy, and we're from Sioux City, Iowa. Hey Robert, all of this sightseeing in DC is making me really hungry. Have you had lunch yet?
No, I'm starving.
I have a peanut butter and jelly sandwich. Would you like half?
Oh, I can't have that. I'm allergic to peanuts.
Oh, I had no clue.
Do you want some whole wheat cookies instead?
Do these have peanuts in them?
Yes, peanut butter cookies are my favorite.
I can't have that either, but thanks. It really sucks being allergic to peanuts. I'm always smelling my food and reading the labels so I don't get rashes all over my mouth and my throat. Millions of Americans have the same problem. So I wonder, if we go to space, will we still suffer from peanut allergies? Since our body functions differently in microgravity, will allergic reactions change?
This is what we already know. Astronauts are more likely to develop infections in space. Wounds heal poorly in microgravity. Scientists speculate that the immune system is suppressed in space. Allergies are a type of immune response. Our hypothesis is that allergic immune responses will be suppressed in space.
So let's take a look at how allergies work. First, the peanut protein enters the body. Next, APC, or antigen-presenting cells, will take up the allergen and present it to a subset of T cells called Th2 lymphocytes, which are represented by ninjas. The Th2 is therefore activated and produces a cytokine called IL-4. The IL-4 will activate B cells, which begin to produce a large amount of antibodies called IgE. Next, the IgE will bind to specific receptors on the mast cell. When the allergen enters the body again, the peanut protein will bind to the IgE on the mast cell. This binding will cause the mast cell to release multiple types of inflammatory chemicals, one of which is histamine. This histamine triggers the allergic symptoms we observe, such as sneezing, itchiness, and others. So our first question is, would the T cells and B cells be activated in space to produce IgE? To test this, we would need to bring live blood to space, which we couldn't do. So our second question is, would the antigens bind with the IgE in space? Without this step, the mast cell will not be triggered to release histamine.
This is our experimental design. We will be using a Type 3 FME mini lab. In the first compartment, we will be putting 2 microteeter wells. One well will be coated with peanut-specific IgE antibodies. The other well will be coated with soy-specific antibodies. This second well acts as an experimental control, so we can make sure that the peanut allergens are only binding with the peanut antibodies in microgravity. In the second compartment, we will put solution containing peanut proteins. The third compartment will contain a blocking buffer that will stop all reactions so that there will be no more binding when the FME enters back into Earth.
So how does the ELISA test work? First, the well is coated with antibodies, in this case, peanut-specific IgE. A solution containing antigens is put into the well, in this case, peanut proteins. After the antigens are allowed to bind with the antibodies, a conjugate solution is added, which contains an antibody which will also bind to the antigen. This second antibody is linked to an enzyme. Finally, a substrate solution is added, which will cause the enzyme to produce color. To analyze the results, an ELISA color reader is used to measure the color intensity. The more intense the color, the more binding that has occurred. We came up with 3 possible outcomes. The first outcome is the color intensity of the well from space is the same as the color intensity of the well on Earth. This means that microgravity has no effect on the binding of peanut allergens with peanut-specific IgE. The second outcome is the color intensity of the well from space is the same as the color intensity of the well on Earth. So this means that peanut-specific IgE and peanut protein bind more in microgravity. This means that allergy symptoms would be more severe in space. This is a very serious matter because astronauts with mild allergies, such as itchiness, may develop fatal reactions in space like suffocation if exposed to the allergen. In this case, NASA will have to take all precautions to have no traces of peanuts in the astronauts' food. The third outcome, which is our hypothesis, is the color of the well from space is less intense than the color of the well on Earth. This means that peanut-specific IgE and peanut protein bind less in microgravity. If this is true, it is possible that if we simulate microgravity, that is a feasible way to relieve peanut allergy sufferers here on Earth. That is great news for many people, including myself. This study also brings up another question. Do vaccines work in space? Vaccines stimulate production of antibodies released to our blood to combat diseases. If the antibodies lose their ability to bind to microbes in microgravity, astronauts are vulnerable to microbial diseases. If one day humans would live in space, we would have to come up with other ways to prevent diseases because vaccines might not work. I'm very anxious for our results, but I guess we have to wait longer now.
We thank the following organizations and people for their support: CASIS, Sioux City Community School District, April Tidwell, and special thanks to Dr. Gail Bishop from the University of Iowa.
We need to get an award for most creative presentation. I thought that was incredible. Okay, let's thank Sioux City one more time.